back, friends. Welcome again in another video tutorial from Microbiology with Shagnik. So in that video tutorial, or this video will going to be the third video lecture of gene therapy. In previous two lecture video we have discussed about first one we have discussed about the introduction of the gene therapy, where I have told you that gene therapy is a newly approved or newly developed treatment where we are using gene or oligonucleotide, stretch of short oligonucleotide as a therapeutic compound instead of using the normal drugs. Okay, but mind it very carefully that gene therapy is only applicable to treat those kind of disease which is due to some faulty genes. Okay, clear? Because with the help of gene therapy we will only insert by the gene cloning method the foreign genes which will either replace the faulty genes, or they will produce some kind of proteins which will causes the correction of those faulty genes. Okay, in various kind of way. And in second video we have discussed about two type of gene therapy. One is the somatic gene therapy, in which I have told you that this kind of gene therapy simply targets the somatic cells. Okay, and they are not heritable. I hope you can understand why, because somatic cells are not involved in any heredity, right? And another one is germ line gene therapy, okay, where we are targeting the germ cell, especially eggs and sperms. Okay, so the second kind of gene therapy is heritable. So now today we will going to discuss about various kind of gene therapeutic strategy. So I will going to discuss with you about two mechanisms by which we can use gene therapy to treat various kind of problems. Okay, so the first one is the gene augmentation therapy, or commonly known as GAT. So let's see what is going on in GAT. So in GAT we are simply adding a functional allele, okay, which will treat any kind of inherited disorder that is due to deficiency or genetic deficiency of the gene product. Okay, that means let's try to understand. Suppose a genetic makeup is small t, which is responsible for a particular type of disorder. I hope you can understand that it is a autosomal recessive disorder I am talking about, right? So under such circumstances, if we use GAT to insert a capital T, so what will happen? According to Mendelian principle, then the capital T will expressed, and as a result what will happen? The expression of the small t will be masked, and that is how GAT help us to treat autosomal recessive disorders, okay, by simply adding a functional allele, okay, that will treat any kind of inherited disorder that is due to genetic deficiency of a gene product. Okay, so let's see how it is going on. So we will add the allele in the disease cell. Okay, so as a result they will get back their normal phenotype. Okay, so remember one thing very carefully, that we can use GAT for autosomal recessive disorder treatment, but we cannot use, or we can use but we will get very low result when we will use them against any kind of dominantly inherited disorder. Okay, I hope you can understand, because if there is a capital T, if you add a small t with the help of GAT, that will not going to solve your problem, because capital T will still be expressed, right? That is the idea. So now next let's talk about the next important principle, that is targeted killing of specific cells. Okay, how it is done? So here we are simply utilizing a gene, or we are simply utilizing two kind of gene depending on our choice. One gene will code for a specific toxic compound. Okay, so that is why this gene known as toxic gene, or more specifically and importantly suicide gene. Okay, so we are transferring those kind of gene in the cell okay, for which it is extremely toxic. So now the question is, why we are inserting a toxic gene which is toxic for a particular cell and we are telling this process as a part of gene therapy. So let me introduce, there are various kind of cell, or more specifically saying there are various kind of transformed or transfected cell which are commonly known as cancerous cell. And is they are good for our normal health? No. That is why we are using that destructive property of this gene to destroy those cancerous cells. Okay, so this gene will be transferred to those kind of cells, and in the cell the gene will encode the toxic protein and cause the destruction of the cell. Not only cancerous cell, any kind of transformed or transfected cell which can be a threat for our normal body can be destructed with the help of this kind of gene therapy. That means by using gene therapy we are not replacing the gene, or we are not just correcting the gene, but with the help of gene therapy sometime we can also destroy the cell. Okay, so that is how it is going on. Upon addition of the toxic gene in the cell, they will facilitate the destruction of the cell. Okay, another important mechanism, that is when we will use a gene encoding prodrug. Okay, so these prodrugs are nothing but the reagents that will confer sensitivity towards the subsequent treatment with a drug. Okay, that means what? This prodrug along with drug facilitates the destruction of the cell. Okay, so again prodrug is a reagent that confers sensitivity to the subsequent treatment with a drug. Okay, so that is how we can also ensure the destruction of the cell. Okay, so in the second method we are simply destructing the cell by using two procedure, right? In one procedure we are using toxic compounds to facilitate that extraction, and in another procedure we are using simply prodrugs, right, that along with drug causes destruction of the cell. Okay, and in the first type of strategy, that is our GAT, that is gene augmentation therapy, we will add functional allele. Okay, this functional allele will treat any kind of inherited disorder that is due to genetic deficiency of a gene product. Okay, so GAT is mainly applicable for autosomal recessive disorder. Mind this very carefully. We cannot use GAT for any kind of dominantly inherited disorders. Okay, so that is all about gene augmentation therapy and targeted killing of specific cell. That is the two kind of strategies that gene therapy use. Okay, so that is the end of that video. So I hope this video will be helpful to you to understand the basic concept of the gene therapy strategy. So we will again come back with another gene therapy video where I will going to start about the ex vivo and in vivo gene therapy. So till then, bye bye friends.